In this video, we're going to be talking about the Krebs cycle. So let's not waste any of your time. Let's get right to it. Now, when we're thinking Krebs cycle, okay, what we want to think about is really getting into the breakdown of that molecule, that glucose molecule. But we also want to think about, and you can see it in the diagram there beside me, is that we've got a circular pathway. Okay, glycolysis, if you remember, was a linear pathway. In other words, it had a start and a finish, and they were two different locations. If you think about a race, you know, where you have a starting line and then your ending line is just a straight line, you know, 5 miles away or whatever the race is, a mile away, and that's where the finish line is. That's a linear pathway. A circular pathway, as you can see, this would be if like at a track meet where you're doing the 400, and where you start and where you end is the same location. And there's a couple of benefits either way. One we're going to focus on is, and we'll see this when we're talking about it, especially here, and especially in class, is that the main difference in this case, the reactants and the product are the same in a circular pathway, but are different in a linear pathway. In glycolysis, we started with glucose, and we ended up with pyruvate oxidation. But what we're going to see in this process, because it's a circular pathway, is that from a efficiency standpoint, when the process ends and it gets back to the top of the circle, you end up with the same molecule as you started with. In other words, that allows the mitochondria, or the cell, to just keep cranking out this process. So while you're working on a molecule, the next molecule is arriving, and we can go right into the next steps. In other words, this can be a continual process. Glycolysis is dependent upon having the next glucose molecule, and it starts that change. But this Krebs cycle, it ends with the product that's needed for the next go around. So a pretty efficient process. The takeaway here is that you can see at the bottom you're making ATP molecules, but the big takeaway is all of the NADHs. Okay, you make 3 NADHs per molecule, and remember there's 2. So when we look at our end product, we're going to be making 6 NADHs. Okay, so we're really cranking up the electron carrier process. But we also make an FADH2 molecule, and we release a couple of carbon dioxides as well. So, but again, we always want to think about this stuff in tandems. If we're releasing or removing H's from the glucose molecule, it's being oxidized. The NADH is being reduced, as well as the FAD. Okay, if we're making an ATP molecule, then we saw an example of that enzyme with the ADP attached to it, and the molecule that bringing the phosphate being attached to the enzyme as well. We saw those examples. But the idea of the acetyl-CoA, okay, energy inside that, but again, making those electron carriers, making those reduced molecules, and just the idea of when this product is, or when the cycle is complete, we've got the next molecule needed for the next process, and we'll talk about this heavily in class. But just the idea of circular versus linear, and what the main benefit of each of those are. continuing to think about the Krebs cycle. Now, those acetyl groups that we made in pyruvate oxidation, okay, are oxidized to produce, the electron carriers. We're taking the NAD and adding those H's. We're taking the FAD and adding those H's. Glycolysis was a 10-step process. This is a 9-step process that really begins with the acetyl-CoA being bonded with the oxaloacetate, which is the final product of Krebs cycle. And that product is in as match with the next acetyl-CoA. And when they do, they make a citrate, which is your first conversion within the Krebs cycle. But again, it ends with, that's what the regeneration of oxaloacetate refers to. When the cycle reaches the end, we're back to the end product, which is connected to the next acetyl-CoA that comes through. And of course, as we've mentioned many times, we've seen examples. Enzymes do all of this. And when we're in the classroom, we can look at the steps individually out of the textbook, and they will list the enzymes, and we'll talk about that process as well. All right, so looking back at our diagram, okay, and looking at some of the end products, okay, we make 2 molecules of CO2 for each acetyl-CoA. So that means when we look at our end products, we're going to see 4 CO2s. For each acetyl-CoA, we make 3 NADHs, so we're going to see 6 on our end product. Each one makes an FADH, 2, so we're going to see 2 on our end product. And down at the bottom, we produce 1 ATP molecule. So on our end products, we're going to see 2 ATP molecules. And of course, it ends with that oxaloacetate. And again, but we'll talk about that in class, and we can look at the diagram in full on the screen or however. But just the efficiency of that ending and then going into the next one, that's the efficiency of a circular pathway. Alright, so here's our summary screen. The purple box, I mean, we've seen the diagram, we've seen the acetyl-CoA, what they each make. And don't forget, we're in the Krebs cycle, so we are deep into the matrix of that middle part of the mitochondria. But when we're looking at end products, everything so far has been 2. Glycolysis was 2-2-2, 2 pyruvates, 2 ATP net, and 2 NADHs. Pyruvate oxidation, again, was 2 NADHs, 2 acetyl-CoA enzymes, and 2 carbon dioxide released. Okay, but now we see some different numbers. So one thing to think about on your test or on your quizzes, if you see a 6 or a 4, okay, for an end product, okay, you want to be thinking Krebs cycle. Okay, Krebs cycle. The other one that could also be a 4 is if the question asks you how many ATP molecules are made during glycolysis. That number would also be 4. But for the most part, you're asked on that is what's the net gain of glycolysis. So, but if it's a number different than 2, for the most part, you can think Krebs cycle. Okay, because you got 6 NADHs that are made, 2 FADHs, 2 ATP molecules made, and then 4 carbon dioxide molecules are made. Excuse me. But again, just straight memorization would be the way to go with that, because you can remember the numbers, but you can't. But if you don't memorize them, you don't always remember the products and the product names. So, but that's the idea with our summary slide for Krebs cycle. Now on this slide, to this point, through glycolysis, through pyruvate oxidation, through the Krebs cycle, glucose has been oxidized to make these 6 CO2s. There were 2 made in pyruvate oxidation, and there were 4 made in the Krebs cycle. We have made 4 ATP molecules. There were 2 in glycolysis, 2 in the Krebs cycle. We have made 10 NADHs. There were 2 in glycolysis. There were 2 in pyruvate oxidation, and now 6 in the Krebs cycle for a total of 10. And we've made 2 FADHs, both of those in the Krebs cycle. Now the energy that we're transferring, okay, the electron carriers, they are carrying all this stuff, all these electron carriers are carrying these H's to the electron transport chain, the ETC. But we got to think energy. Okay, we're going to be making ATP molecules, and we'll see this, but these electrons are going to end up activating the active transport pump that's going to drive or allow the H's to pass through and generate the ATP molecules. So again, here's our purple summary, and you can see that we're building it slowly. Now we've got 3. So again, these are the slides that you're really going to want to pay attention to when it comes time for quizzes and tests, because they have all the information on them. Locations, starting materials, end products, and again, you can see all the 2s on there for glycolysis and the 2s for pyruvate oxidation. And then we get that 6 and a 4 for the Krebs cycle. So these are going to be super important to make sure you know. And the idea is to, hey, just memorize the thing. And then when it comes to questions, you've got it under control. Well, that is our look at the Krebs cycle. The next time we come together, we're going to be looking at the electron transport chain. We'll see you then.